let's continue on with our unit. We are now here at the very end. So basically, we're going to learn how to read a karyotype, and we're going to be able to just identify more genetic disorders. So just to review and reflect, remember diploid and haploid cells. Remember diploid was with mitosis, and it was 2 cells, and haploid was in meiosis, and it equaled up with 4 cells. But when diploid happens is when egg and sperm meet as well. So the body of the cells in plants, the chromosomes occurs in pairs. As we remember when we talk about meiosis, because this is our thing for today is meiosis, a male parent and a female parent. Obviously, that is egg and sperm. So diploid and haploid. So diploid, remember this together. Haploid is separate. Remember, a human has 46 chromosomes. So diploid cells contains each kind of the chromosome, which is again written as 2n. Haploid is half, so it's one of them. The chromosome number is of a species is not related to the complexity of the organism. Just because we have 46 doesn't mean we are the most complex. But just because a pea plant has 14 doesn't, again, it's not related to the complexity of the organism. Remember, homologous chromosomes review. So this right here means same. So these are the same pairs. And remember, they're each one are having the same trait in the same order. They're not always going to be identical because that means everyone has different alleles and different genes, right? We're going to connect the I genes together, but does not mean that you would be the same color I genes that you get. One's going to be dominant and one's going to be recessive. And then, obviously, the dominant will win. So let's go back over meiosis. When cells divide by mitosis, the new cells have the same number, right? But meiosis, remember, it is half the number. So if we start with 46, we're going to end up with 23, okay? This is just review. So we remember talking about sexual reproduction. So, of course, sexual reproduction is the fusion of haploid cells, which is your gametes, your egg and sperm, okay? Sperm is 23 chromosomes. Egg is 23 chromosomes. When they come together, they make a zygote, which is 46 chromosomes, which is now considered diploid. How I know it's diploid, not only because it's 2n, but diploid would only have the full number. So sex determination. So remember, girls, we do have 46 chromosomes, 23 pairs. And the first 22, we're going to read about a karyotype today. The first 22 are autosomes, which are body cells. Okay, number 23 is sex cells. Okay, those are called sex chromosomes. Remember, girls and boys. Girls is XX, boys is XY. Y determines the gender. Why does Y determine the gender? Because if you have a Y, you're a boy. I don't care how much a girl you look like. If you got a Y, you are a boy. If you do not have a Y, I don't care how much of a man you look like, because you don't have a Y, you are a boy. So remember, meiosis begins with the diploid cell, and then it splits, and then it ends up splitting again into 4. So it starts off as it begins with diploid, and then ends with haploid. But there's some mistakes, and this is one thing that we did talk about before, is non-disjunction. This is when they fail to separate. So if you look here at this picture, what's supposed to happen? This right here was supposed to separate. It wanted to stay with its sister, and it's not supposed to do that. It's supposed to grow up and pull apart. And so this is, again, the cyclin malfunction. Remember the cyclin? That is the enzyme. So this is called trisomy, this is called monosomy, and this is called polyploidy. So here is trisomy. So trisomy is what we're going to consider with Down syndrome. Okay, this is when we get an extra chromosome. That's how we get for, like, the sperm here was 23 and mom had 24. Her child came up with 47. That usually happens here on number 21. Number 21. Not saying it can't happen anywhere else, but because we're specifically talking about Down syndrome usually happens on number 21. Okay, on number 21. And that is called trisomy. Why tri? Tri means 3. So 1, because you see everybody has a set of 2, right? This one has 3. It's just like you and your best friend going on a date, but you have that third wheel. That's trisomy. So just look at the characteristics. So you look here, you notice that everybody who has Down syndrome, I don't care what race you are, you look alike. the eyes, the face structure. Even look at the tiger. Really look at him. He looks just like any other person who got Down syndrome. Mistakes in meiosis. Monosomy. Mono means one. So what happens here? Well, that the guy shot out a blank. And so it still fertilized the egg, but the girl only got one X. So this is when we're missing a chromosome because now we were supposed to have 46 and now we're going to have 45. This example will be like Turner syndrome. This is when we're missing an X chromosome on the 23rd pair. So Turner syndrome. So 1 out of 5,000 live births, most of them die, okay? There's reasons why. They only viable monosomy in humans. Only in humans, okay? Women only have 45 because remember, women supposed to have two X's. That little circle there is a blank. It was a sperm that fertilized, but it was a blank. They are genetically female, but they do not mature sexually during puberty and are sterile. So yes, they are genetically female. They have a body part as a female, but they do not have a uterus. They do not grow breasts. So they're kind of different. Short stature, they very normal. It's just they would never go through mature sexually, so they will stay really little. Here's an example of a picture right here. As you can see her neck, how her body's shaped. She's kind of boxed off a little bit, and that is because now this right here is what I was talking about. This is a karyotype with these little lines. This is the chromosomes. So the last two is number 23. As you see, most of them are labeled. They usually go big to small, and then all of a sudden you have a big one. This is how you know. 1 through 22, remember, is body cells. Look at number 23. It's supposed to be 2. She's only got one X. So polyploidy, poly means what? Many or more than 3. So this condition actually is when you have way more chromosomes. This is definitely rare in animals because it's lethal. You'll pretty much die if you have that many. But it's definitely happens in plants, and they're usually larger and healthier. You ever had the strawberry and it had like 2 butts and all that other stuff? Yeah, it had a lot of extra cells, and it's actually healthier. It can be triploid or tetraploid. Now, changes in the chromosomes. Remember, the karyotype is the chart of chromosomes. Again, 1 through 22 is autosomes, and number 23 is sex. Metaphase, the chromosomes are enlarged and cut apart and arranged on this chart. So in metaphase, they are enlarged, cut apart, and arranged in pairs. And according to their length, that's where we put the location of the centromere. Now, how do you know if your child has Down syndrome, or how would you know that your child may have some type of disease, or you want to check the genetic analysis of your baby before he gets here? You do an amniocentesis. Amniocentesis is basically a big old long needle that goes through the woman's stomach and goes into the embryo and gets some fetal cells, and so we can actually go ahead and start diagnosing the child. So as you see here, we have a long needle and it goes in, and then once it goes in, they have the ultrasound to make sure the needle doesn't hit the baby, but they just get a little bit of cells. This is always a 50-50 chance. The reason why it's 50-50 because sometimes this will not close, and so the baby, it leaks the fluid and the baby will die. Sometimes the baby is punctured. But this is a normal karyotype. As you see, it goes from big to small, and then at the very end, it is X and Y. A lot of times it may not even say X and Y. You just have to recognize. So a girl, her X's are long. Y's are little. So by looking at this, it has an X and a Y. This is a boy. And he's normal. I don't see any extra chromosomes, right? We just see 2, 2, 2, 2, 2. So this is if you have Down syndrome. So it was like, okay, 2, 2, 2, 2, 2, 2, 2, 2, 2. Everything looks normal. But all of a sudden we get to number 21. We had 3. Remember, that's called trisomy. But is this a girl or boy? This is a boy because it has an XY. Now, Turner syndrome. So Turner syndrome, we're looking, okay, good, good, good. All of a sudden, we keep on going. Okay, 1 through 22, you all got a partner, but she doesn't have a partner for number 23. Remember, that is called monosomy. And now this is like of a strawberry. What's wrong already? I'm just using an example. This is actually from a human or animal because it does have a sex chromosome. But if you look here, 1, 2, 3, 4. No, we only supposed to have 2. So something's not right. We got 3 here. We got 3.5. We got 2. I don't know what those are. This is polyploidy. Honestly, this is kind of how some of your cells look like too when they begin to go under cancer, because it begins to rejuvenate and it begins to make bad cells. Some of your chromosomes begin to look like this. And of course, this has 2 long lines though. Whatever this is, we do know it is a female, but we know the female is obviously something's not right. There are environmental factors that can change this. We have certain DNA environmental factors like changing DNA sequences and changing amino acids and things that alter proteins, tobacco use, too much UV, poor diet and exercise. So here, look at this chart, and this is a cause and effect of environmental stuff. This can change and mutate your genes. So just review. Mutations that occur in sex cells can be passed from parent to offspring during what? Sexual reproduction. So as the egg and sperm come together, it can be transferred. Why does meiosis promote genetic variability? Because it goes to cell divisions producing haploid cells, which gives us genetic diversity. Trisomy on chromosome 21 is probably caused by non-disjunction, where they did not split during metaphase to anaphase. Because when the anaphase pulls apart, the spindle fibers didn't connect to metaphase, and anaphase it just it went with the sister. But other than that, that is all.